Genomics. What is genomics? Genomics is the study of structure, function, evolution, mapping and editing of genomes, where genome is the complete set of DNA, including all its genes, in an organism. Genomics, the study of an organism's complete set of genetic information. Genetics, the study of hereditary. The genome includes both genes, coding, and non-coding DNA. The study of function and composition of single genes. Genome, the complete genetic information of an organism. Gene, specific sequence of DNA that codes for a functional molecule. History. Genomics is a concept that was first developed by Fred Sanger in early 1970s, who first sequenced the complete genome of a virus and of a mitochondrion. In 1972, Walter Fires and his research group became the first to sequence a gene. They sequenced the gene of bacteriophage MS2. In 1995, Hamilton O. Smith and his team became the first to sequence a genome of a free-living organism, that of Haemophilus influenzae. Technologies in genomics. DNA sequencing. The process of determining the exact order of the nucleotides, A, T, G, C, in a DNA molecule. Gene editing. It is a process of insertion, deletion, modification, or replacement of DNA sequences. Transcriptomics. It is the study of transcriptomics, i.e., the complete set of RNA molecule. Proteogenomics. Proteogenomics is an interdisciplinary field that integrates genomics, transcriptomics, and proteomics to improve understanding of how genes are translated into proteins. Computational genomics. Computational genomics is the use of mathematical models, statistics, algorithms, and computer science to analyze and interpret genomic data. Branches of genomics. Structural genomics. Mapping and sequencing. Functional genomics. Understanding gene regulation and expressions. Comparative genomics. Comparing genomes of different species. Epigenomics. Studying chemical modifications to DNA that affect gene activity. Metagenomics. Analyzing genetic material from environmental factors. Pharmacogenomics. Tailoring drug treatments based on individual genomes. Application of genomics. Human health and medicine. Agriculture and food security. Evolution and anthropology. Forensics and security. Environmental genomics. Structural genomics. Structural genomics seeks to describe the three-dimensional structure of every protein encoded by a given genome. This genome-based approach allows for a high-throughput method of structure determination by a combination of experimental and modeling approach. Objectives. Solve representative structures from all protein families. Provide structural information for functional annotation of genomes. Understand protein folding and interactions. Develop new drugs by identifying target sites. Functional genomics. Functional genomics focuses on the dynamic ASPE such as gene transcription, translation, and protein interactions. Functional genomic projects, such as genome sequencing projects, to describe gene and protein functions and interactions. Techniques. Transcriptomics. Transcriptomics studies measure gene expression at the transcript or RNA level. Proteomics. Proteomics approaches focus on which proteins are expressed in a biological system but may also include studies of protein structure. Metabolomics. Metabolomics is the study of all metabolites in a biological system. Interactomics. Interactomics is of specific relevance to agriculture systems, particularly in understanding disease. Interactomics is the study of the molecular interactions and encompasses host-pathogen interactions. Epigenetics. Epigenetics studies non-heritable changes in the genome, e.g., DNA methylation and histone modification, that affects observed phenotypes. Nutrigenomics. Nutrigenomics focuses on understanding how diet affects gene expression. Comparative genomics. Comparative genomics is the field of genomics that involves comparing the genomes of different species, or individuals within a species, to understand similarities, differences, and evolutionary relationships. Approaches. Genome alignment right-pointing arrow compares DNA sequences across species. Phylogenetic analysis right-pointing arrow builds evolutionary trees. Synteny analysis right-pointing arrow studies conserved gene order on chromosomes. Homology analysis right-pointing arrow finds orthologs, same gene in different species, and paralogs, gene duplications. Epigenomics. Epigenomics is the study of the complete set of epigenetic modifications on the genetic material or a cell, known as the epigenomics. Two of the most characterized epigenetic modifications are DNA methylation and histone modification. Mechanisms. DNA methylation. Addition of methyl groups usually silences gene expression. Histone modification. Chemical changes, acetylation, methylation, phosphorylation, that affect DNA packaging and accessibility. Chromatin remodeling. Structural changes in chromatin that influence transcription. Non-coding RNAs. Regulate gene expression post-transcriptionally. Metagenomics. Metagenomics is the study of the collective genomes of microorganisms, bacteria, viruses, fungi, archaea, present in an environmental sample, like soil, ocean water, or the human gut, without the need to culture them in the lab. Environmental shotgun sequencing, S, is a key technique in metagenomics. A. Sampling from habitat. B. Filtering particles, typically by size. C. Lysis and DNA extraction. D. Cloning and library construction. E. Sequencing the clones. F. Sequence assembly into contains and scaffolds. Types. Sequence-based metagenomics right-pointing arrow analyzes DNA sequences to identify microbes. Function-based metagenomics right-pointing arrow clones DNA into host organisms to test functions, e.g., antibiotic resistance, enzyme activity. Pharmacogenomics. Pharmacogenomics is the study of how a person's genes affect their response to drugs. It combines pharmacology, the science of drugs, and genomics, the study of genes, to develop safe, effective, and personalized medicines. Applications. Clinical practices. Oncology, psychiatry, drug development. Oncogenomics. Oncogenomics is the study of the cancer genome, involving identification of oncogenes, cancer-causing genes, tumor suppressor genes, and mutations driving cancer. Proteomics. Introduction. Proteomics is the large-scale study of the entire set of proteins, the proteome, expressed by a cell, tissue, or organism at a given time. Since proteins are the main functional molecules, enzymes, receptors, hormones, structural proteins, studying them helps us understand actual cellular functions, not just genetic potential. Types of proteomics. Expression proteomics right-pointing arrow compares protein levels under different conditions. Structural proteomics. Right-pointing arrow studies 3D structure of proteins and complexes. Functional proteomics. Right-pointing arrow investigates protein-protein interactions, signaling pathways. Objectives. Protein, peptide separation. Identification and characterization of resolved proteins by MS. Data analysis and applications. Techniques in proteomics. 2D gel electrophoresis right-pointing arrow separates proteins. Mass spectrometry, MS. Right-pointing arrow identifies and quantifies proteins. Protein microarrays right-pointing arrow detect protein expression and interactions. X-ray crystallography, NMR. Cryo-M right-pointing arrow structure determination. Yeast 2 hybrid system right-pointing arrow protein-protein interactions. Thank you.